Hello everyone, this is Zeshan Shoaib and today I will talk about the variability in response to transcranial direct current stimulation of the motor cortex. This article was published in Brain Stimulation Journal, which is a top 10 journal in the Neurosciences category and whose impact factor is 9.184. In this article, Sarah discuss the variable human response to TDCS. In electrical stimulation studies, we can also face this type of effects. So we can refer this article to discuss the results. In this study, they investigated the effects of TDCS over the motor cortex. This slide will give you the background of transcranial direct current stimulation. So here we can see that transcranial direct current stimulation is a portable and wearable brain stimulation technique that deliver low electric current to the skull. Fixed current between 1 and 2 milliampere is typically applied in these studies. It is thought that TDCS changes the excitability of neurons in the brain by hyperpolarizing or depolarizing their membrane potential. However, despite the ever increasing number of studies using TDCS in fields from cognitive neuroscience to rehabilitation, there are few studies that have been reported the variable effects of TDCS as well. So the question is, how many participants can fail to respond to TDCS, or in other words, how many subjects can be found to change the excitability due to TDCS? In this article, we will try to find the answer of this question. In this article, the authors used 53 right-handed subjects to determine the variability of TDCS effects among the subjects. They used two different modalities. One is the TMS and second is the TDCS to record the brain functionality. So they used a single pulse TMS with the connected figure of 8 coil with internal ring diameter of 7 centimeter. The hotspot was identified as the position where most stable motor evoked potential was elicited, with the coil held 45 degrees to the midline, tangentially to the skull and the handle pointing backward. And the TDCS they applied at the motor cortex. The one electrode was placed over the right orbit and the other electrode was positioned over the previously marked hotspot, and they used saline soaked surface sponge electrodes to deliver a 2 milliampere current intensity over a period of 10 minutes. So in this study, the subjects were enrolled in the experiment with two randomized ordered sessions, anodal and cathodal, with the gap of 3 days in each session. Uh, and in each experimental session, the motor hotspot was firstly determined at the intensity, eliciting motor evoked potential sizes with about 0.5 to 1 millivolt, followed by measurements of motor thresholds, and after latency measurements followed by a 10 minutes break as a measure of baseline motor cortical excitability, 30 motor evoked potentials with posterior anterior directed current during rest was recorded, and after application of either a nodal or cathodal TDCS for 10 minutes, 30 motor evoked potentials with posterior anterior current were recorded every 5 minutes for 30 minutes. So this made 7 time points from T0, T5, T10 up to T30. And importantly, the outcomes of TDCS were completely blind until the end of the experiments from all 53 subjects to the examiner who analyzed the latency of motor evoked potentials with different coil orientations. So in this slide, we can see the detail of statistical analysis they performed in this study. So firstly, they used Kolmogorov-Smirnov test to check the normal distribution of the measurements in the data set. And after that, after confirming the normality, they performed one-way repeated measures of ANOVA with the factor time using non-normalized motor evoked potential values, and to test the correlation between TDCS effects and all measured variables. The responses of anodal and cathodal TDCS were firstly assessed by the grand average of normalized motor evoked potential values, and a regression analysis was computed with the grand average and all of the neurophysiological measurement values, and Pearson's correlation coefficient was also calculated, and they also performed unpaired t-test to evaluate the effects of gender and time of the day, and they used a significance level with p is less than 0.05, and they also performed the cluster analysis uh, uh, in groups according to their individual grand average in response to anodal and cathodal TDCS, and they use the conditions. If the value is greater than 1, it means uh, they consider it as a facilitation, and if the value is less than 1, they consider it as an inhibition. Uh, and they also performed a two-step cluster analysis using log transformed data of normalized motor evoked potential in order to test whether there was any tendency for subpopulations of individuals to react to TDCS in a common way. In this case, the time course of each cluster was evaluated with one-way ANOVA with normalized motor evoked potential sizes. And to analyze all this statistical analysis, data analysis, they used SPSS software in this study. So in this slide, we can see the results they obtained in this study. So figure 1A and figure 1B uh, shows the motor evoked potentials data for all the subjects for anodal and cathodal stimulation. So in these two figures, we can see a large variation in response between individuals. And a one-way ANOVA showed that on average there was a main effect of time in the anodal group, and there was no effect in the cathodal group. Thus, we can say that on average there was an excitatory effect of 2 milliampere anodal TDCS, but not of cathodal TDCS. And figure 1C provides a simple summary of response profile in this population in terms of how many percentage of the subjects responded the anodal or cathodal TDCS. So here we can see that only 36% of the subjects responded to the anodal and cathodal TDCS as they were expecting. Means they showed the increased response in after anodal TDCS and decreased response after cathodal stimulation, 36%, and 38% of the subjects showed uh, facilitatory behavior in response to both of the stimulations, and 21% of the subjects showed anodal inhibitory response to anodal stimulation and facilitatory response to cathodal stimulation. Means 21% of the subjects responded opposite as they were expecting. So this is the overall uh, responses to anodal and cathodal stimulations they found in this study. Thus, we can say that approximately 3/4 of the people had an overall facilitation after 2 milliampere TDCS. They also used cluster analysis to test whether there was any tendency of subpopulations of individuals to react to TDCS in a common way. So figure 2 shows that two main clusters could be analyzed following either anodal or cathodal stimulation. In both cases, there was a group of people who had no overall mean response to TDCS, and they called it a cluster 1 in both cases, and a second group with the mean facilitatory response, uh, and they called it a cluster 2 in both stimulations. So we can see the percentage uh, which did not respond to cathodal and anodal stimulation, and which is 47% and approximately 53% of the subjects. And we can also see the percentage of the subjects in cathodal and anodal stimulation which they respond to the cathodal and anodal stimulation, and which is 53% of the subjects and 47% of the subjects. And figure 2C and D, they plots the mean data from these two groups, uh, and in the anodal session, a one-way ANOVA showed a significant main effect of time in cluster 2 but not in cluster 1, and similar things we can see for cathodal stimulation. For cluster 2, we can see the significant main effect of time but not in cluster 1. They used an intensity of 2 milliampere because it seems to be becoming a standard intensity in the growing literature of TDCS effects on behavioral and cognitive function, and even in clinical settings. And the results were surprisingly variable. After a nodal TDCS, about 3/4 of the individuals showed facilitation and 1/4 inhibition, whereas after cathodal TDCS, the proportions were approximately 60 to 40%, means 60% showed facilitation while 40% showed the inhibition. It is important to note that there is an infinite way in which the TDCS parameters can be combined so that the results of this study may not apply equally well to all the paradigms. And the other limitation of this study is that they did not include a shame condition, and they extrapolate the results from the baseline data. So the conclusion of this presentation is that the effect of TDCS are highly variable, with around 50% of individuals having poor or absent responses. So we can say that if half of the recruited participants are unlikely to respond to TDCS, it will severely hamper attempts to assess treatment effectiveness. In the end, I would like to say thank you to all the viewers for listening me, and hope to see you next time with another video.